We are very privileged to have Dr. Robert Morris here with us from Institute for Systems Biology in Seattle, USA. Dr. Morris conducted this course only one or two places in whole world other than Seattle, maybe in some other part of world. Korea probably you did. It is very first time happening in India, this particular course, and he graciously agreed to bring his entire team with him. So let me welcome all of them here. They're all instructors going to be for this course for next 5 days. And as you can understand, it's going to be a very intense course because 5 days you have to totally learn the traits of the tool. You have to know how you can utilize different type of platform, different type of data sets generated from mass spec and make best use of that for different areas, especially the targeted proteomics field. So it's going to be really motivating, stimulating course here for all of you. And I'm sure that you will have the best time with his team and you will grab the best knowledge available. Additionally, for each day in the evening, we have made some evening innovative sessions. And today being the first day, we will have from 5:30, we have the Human Phenome Project and Metabolomics, a full session based on that, which may not be directly relevant to what you're learning here, but those are independent activities, which we thought are very relevant to update all of you because these things are happening elsewhere in the world and latest in the field. Tomorrow, we are going to talk about biosimilars and biotherapeutics characterization using mass spectrometry, again in the evening. And then on 12th, we are going to talk about biomarker discovery and validation using mass spectrometry. So that will be on 12th evening. These 3 days, evening sessions are going to be very thematic seminars, no hands-on. And last 2 days, because we understand many of you may not have too much basics on the mass spectrometry, especially the data generation part, and some of you are keen to learn that as well. So last 2 days, 13th and 14th, we have also kept some hands-on session in the evening. And that will be based on both QTOF-based proteomics and triple quad-based workflow. And because the numbers are going to be almost 35 to 40 people, so we have made 4 groups, just so that you have the smaller group to do these kind of hands-on workshops. The way this particular thing is going to work, couple of lectures and certain modules for the TPP part going to happen every day. And it means you are going to learn some basics and you are going to apply that on certain modules, do certain hands-on part, do some data analysis part, and then you are ready to write that quiz. Okay, so this entire course you should treat like any course which you are going to learn in any place. And you have to be very vigilant. It's not only just, you listen some lectures. You should try to make as much hands-on as possible. Apply these, take some new data sets, and start analyzing those yourself, just so that then you can become much more confident and comfortable in this area. Additionally, some of you want to continue for the second module of the Gian. That will be core on the mass spectrometry-based workflow. So you can stay for that module as well. And CP Office of IT Bombay continuing education program will be helping you for that part of the Gian part. So with that, let me thank Dr. Rob Morris and team again to really come here with a lot of hassle for traveling, visa, so many other issues. And finally, they're all here and they're going to spend 5 days time with you to teach you this course on Trans-proteomic pipeline module. Thank you very much, Dr. Rob Morris.
Thank you very much. So welcome everyone. Thank you for coming, and we're going to have a number of different things we're going to talk about this week, and there are also some hands-on applications. And so one of the things that we want you to come away with at the end of this is that you know how to use the software, you understand why we're using the software, and you can use those in your research and get those results that you really need to get. So this will be intensive, and we'll have a number of different things. So as Sanjeeva said, this is a connection with the Indian Gian, and this is a new program that came forward and has been very graciously accepted by the government. And so between Sanjeeva and myself, we've put together part of this course. So this will also continue on after this. And then also we're talking about what we can do in the following year as well. So this is a really prestigious component that we're having to go forward with. And so it's excellent that Sanjeeva has been able to get this up and going. So this is the outline of the course that you have. So you've seen that within your documentation. So we will have 5 full days of talks. This will be interspersed. The last 2 days, the Sunday and the Monday, is really where they have the main conference as well. So there'll be some talks backwards and forwards there. But you'll see that we're going to take you through the trans-proteomic pipeline. And so the trans-proteomic pipeline is our bioinformatic process We'll take you through all the very basics of doing database searching, how do you get your data into the program, understand the statistical validation of your results in the very primary efforts, and then starting to understand how you can look at post-translational modifications as well as statistical validation. And so as we go forward, we'll then also talk about some more advanced proteomics, which is the swath or data-independent analysis type approaches. We'll talk about how we link a lot of these things into targeted proteomics, which is a major focus of this conference, is how do we actually do a lot of quantitative proteomics, and we'll look at the resources and the tools that we have for selected reaction monitoring. You may also know it as multiple reaction monitoring. That's something you do in a triple quadrupole. Then how those things interplay between the data-independent analysis, the very advanced mass spectrometry that we'll also talk about. And then finally, over those 2 last days, we'll go forward and talk about some tutorials and actually get you to process some of the data yourself. We'll help you through those. So as you're processing the data, we'll take you through that, and you'll have a look at those results and how we're actually going to go about and do that. So here are your tutors for this week. Apart from myself, we have Luis Mendoza, and so Luis will put his hand up. So Luis is an excellent teacher, and he'll guide you through a lot of the software. He's a developer of the TPP, so he knows intimately the ins and outs of all of the programs. Michael Hoopman is also here. So Mike is also a developer of the TPP, but he's also a mass spectrometrist, a researcher, so he has a vested interest in the software to make sure that it actually works, and he'll talk about a lot of the cross-linking efforts that you'll be also learning about as well. David Campbell. So David Campbell is a developer of the Atlas resources, and so he will talk to you about some of the resources that we have online for when you're starting to compile your data, how you actually utilize things like Peptide Atlas, SRM Atlas. And then Ulrike Kuzerbauch, she's here as well. Ulrike, put your hand up so everyone knows who she is. And she'll talk to you about the targeted proteomics, the efforts that we have to give you the resources to analyze any human protein of interest and show you how you can get that data. And then finally, Sam Bader is also here. Sam was in my group, but he's just recently left my group to go back to Switzerland. But he's been a primary developer of swath for SciEx instruments, the triple TOF instruments. And so he'll take you through not only the data analysis, but also the data resources and what he's developed, the swath atlas in my group. And so how can you utilize those tools to get them to work for your experiments as well. And so I'm just going to give a little bit of an introduction at the moment, just briefly on some of the things like the modern day mass spectrometers, the data collection, and also some quantitative applications, just to give it a bit of a background to what we're actually doing, and then we can get into the meat of the conference and really talk about the data analysis and the data software tools that we have. But to give you a bit of a background, so this is what we call the systems biology trinity of analysis. And so you can see here, we class systems biology to be a component of 3 separate things. One is we start off with a biological question. And that's a question that could come from any area. It doesn't have to be from human. It could be also infectious diseases. It could be parasites. It could be plants. It could be animal health as well. And so it really comes into, so what kind of biological questions do you want it to define? One could be biomarker discovery. Other ones could be things like trying to understand protein-protein interactions, those sorts of things. And then you start to look at what technologies does one have to start to collect data about what you're trying to question. And do you actually have the right technology? Do you need more technology? Do you need technologies that other people may have that you can collaborate with? And then finally, we have also the computational effort as well. And so the computational effort is where you take this information that you've collected from the biological question, you've done your technology, and now you try and analyze that information. And how do you bring that information together so that you can come up with an answer that can then be fed back into answering this biological question in the first place, but maybe give you new hypotheses to which you want to start to ask. And so you go around this continual circle of development. And so It really drives the technology even further, and it tells you the importance of the bioinformatic analysis as well to try and understand the massive amounts of data that you're collecting, not only from some proteomes, but genomes and also metabolomes. You have a vast array of data that you need to understand, reduce the data to something that makes it meaningful, and then to be able to make your discoveries out of there. So that's really what we call the systems biology tool. And on the outside here, I've just placed a number of different areas of which we have different efforts going forward in terms of the systems biology tool. And so these are some of the developments that we have, just particularly in my group. So with the bioinformatic efforts, we have a number of different programs. So we have the Trans-proteomic pipeline, which you'll learn about this week as well. It's a complete software suite to analyze mass spectrometry data. We have the Peptide Atlas, which is the compendium of the results, and then we'll go through those. The SRM Atlas, which is a tool that is a compendium of all the fragment or transitions that you need to program a mass spectrometer to target any particular peptide. We have PASL, which you can upload your data. So if you're publishing SRM or MRM data, you want to upload your data here so the reviewers can actually see that, and you publicly release your data. And so that's one of the big things that we have going at the Institute of Systems Biology is to make all of our data and all of our tools publicly available. And we encourage people to do that as well, to make their data publicly available, because your data, beyond the question that you've asked, has a number of different lives after that, because many other people may want to work on the same system and they would like to see what other people have already done, and maybe they can use your data there as well. And so it really pays to do that. We have other areas as SRM Quant Atlases, which we try and define what the limits of detection, limits of quantitation are for targeted proteomics. SWOT Atlas, which we'll talk about, is where we have a repository of spectral data for data-independent analysis. And as of last year, we already started a collaboration with some people at NII, and this is now being finalized at the moment. So we'll have the Indian Rat Atlas going into the SWOT Atlas, and so Sam will talk a little bit about that. We have some tools as well which is recently developed, the SWOT Prophet. We'll talk a little bit about those, and then finally we'll also talk about cross-linking applications. So if you're doing some very high throughput protein cross-linking, this is the type of software that you'd really want to get hold of, and understanding how to use that is a real benefit. So this is where we are at the moment in terms of mass spec-based proteomics. So we have a number of different tools that you or you may have heard of, or probably a lot of experienced in. The first one is shotgun mass spectrometry. So shotgun mass spectrometry is you put your sample on the mass spectrometer, it programs the top 10, top 20 ions that it wants to see, and then fragments those. Then you do a database search and you end up with a list. And so that gives you a capability. You get a lot of proteins being identified, but it doesn't really go very deep into the proteome at all. And so over the last couple of years, there's been this development of trying to broaden that depth of analysis into a proteome. And so by using directed or shotgun using inclusion lists, you're able to then avoid the mass spectrometer only going for the most abundant peptides, but going for peptides that are least abundant, and now you start to broaden out your identifications. But the problem is you start to lose out in terms of all of the proteins you're able to identify at the high abundant level. The third step was to go away from any sort of instrument decision making for your analysis. And so this is where you no longer tell the instrument, just pick whatever peptide is there as most abundant, but you actually do things now that are targeted, so that you're looking specifically for a fragment, and then looking specifically for those ions in a chromatographic peak. Lastly, the data-independent analysis is becoming more and more important these days. We have what we call the SWOT analysis, which is doing a very broad cut of all of the peptides going into the mass spectrometer, fragmenting those, and then we have tools to deconvolute those fragment ions to get back to a chromatographic peak and do that quantitation. We'll talk about that. And so where we see the future, so we see the future in proteomics as being very comprehensive, sensitive, and also reproducible. And so we're going to get a very broad coverage of the whole proteome that we're actually seeing there, rather than just these very shallow cuts. And then ultimately, getting beyond this comprehensive analysis is also bringing in protein modifications in which we can start to determine all of the peptide modifications that are present either by post-translational modification or some other chemical modification that's actually there, and by using this data-independent analysis and getting a complete picture of what's really going on. And so these are giving a number of different advantages, but we also have a number of different challenges that we need to work on to continually get this methodology up and working. So the reason why we're doing all of this is because we understand that the genotype doesn't necessarily reflect the phenotype, and so we have to understand from what we know about the genome, how does that translate to transcripts and then ultimately proteins and protein-protein interactions and also protein modifications. So from the first outset with DNA, from the year 2000, we're able to get a comprehensive part list from the human genome analysis. And so that would enable us to predict which genes are actually being expressed and also which control regions there are for those. With the advent of microarrays and trying to understand the whole repertoire of transcripts, we could actually see which genes were being expressed at what time and at what abundance, and that gave us a quantitative map over this dynamic view of RNA and how these were changing over time. And that enables us to give these predictions in network inferences and also small-scale dynamics. But really, we need to do this at the protein level. The transcript is not a proxy for the proteins. The transcript is an analysis point which can tell us what genes are being expressed and then also how many of those can actually be translated into proteins. But the ultimate goal is to understand the protein, what it's doing at that specific time, at that spatial place as well. And so we need to provide these comprehensive tools to give us this quantitative analysis and view of the protein abundance and its interactions, because there's also things like organelles that could contain no transcript machinery or any protein production machinery, but yet their cargo is primarily protein, and you need to understand what those proteins are doing and how they interact with different cells. And once we get able to do that, we can then understand this behavior of the system beyond just the transcriptome. So this is really what we're trying to understand as well, and the two different areas of which transcriptomics and proteomics has been able to compete with. And first of all, if we're giving a set of protein measurements, we can understand by which proteins are being expressed at any particular time. We know that there are protein interaction databases we can bring together and understand how they're all connected, and also look at perturbations, in which we can see the dynamic changes as we go through. If we make these perturbations, we can understand the differences between two phenotypes and then predict which is actually occurring at that point in time. Now, transcriptomics has a capability of giving us a lot of information that's actually there, but it can't give us information on terms of protein interactions or protein signaling networks and the way that they're directed as well. This can only come from proteomics, but we can combine these two. We can really understand what happens at a cellular network. And this is where proteomics is a unique tool that needs to be part of your repertoire as we go forward. And so in the end, we get this holistic picture of what's occurring both at the transcriptome and the proteome level to try and understand phenotypes. So how do we do that? First of all, we really need to understand what's actually in the system. And so there's a number of different efforts that have gone on in the past to try and define what we have in life. We know this from the periodic table, trying to define all the elements. And then more recently, as we're trying to survey all the proteins, we can see this is almost like a Google-type map of the whole world, which we can then catalog all of the proteins and different components of the cell. We can catalog all their signatures to try and measure those until we get to a point where we can understand which proteins are being expressed under what structures, how they are coming together in terms of modules or structures of different proteins to create protein machines, and then how they actually affect the cells and then also their partners as they're moving around in the body. So how do we do that? So with proteomics, it's a fairly simple type process. you have to start with a sample in the first place, and you have to get that into a mass spectrometer. Usually you can either fractionate your protein in some way, or you do some other isolation technique. But what you end up with is a purified liquid form of the protein. You have to do some hydrolysis, or whether it be either trypsin or some chemical cleavage point in which you can make those peptides shorter. But these are also techniques that are being worked on so that you can do them with larger peptides as well. But what we're going to talk about this week is mainly this process of converting into small peptides. And then we introduce them into a mass spectrometer. So we want to try and introduce them in the most efficiently way as possible, so we get the whole fraction of the peptide complement going into the mass spectrometer so we can analyze that. And that is somewhat of an art in itself, and really hasn't changed in the last 20 years. It's really begging for new ways of optimizing the way that peptides go into the mass spectrometer. And then finally you have what we call the database search. And the database search is what some people might see as a very simple component, but it actually is quite complex in trying to understand all of the fragmentation profiles and doing statistical validation, so you know that the peptides that you've identified is actually correct. And then also you can create databases in which you can use that data for your research. And so in terms of modern day mass spectrometers, so we've got 20 minutes to go. So I'm just going to go through this briefly. There's a number of different types of mass spectrometers. There's really two classes. One's called tandem in time and also tandem in space. And really, when we talk of tandem in time, we're talking about trapping type instruments. Either ion traps have been a great boon in the proteomics era for the last at least 10 to 20 years, and has given us a lot of data. There's also these Fourier transform ion cyclotron resonance mass spectrometers, FTICRs, which also gave us some very early important data in high resolution, high mass accuracy, but have fallen out of favor for things like the Orbitrap, which doesn't require the resources to keep these things going, like the helium. But in terms of tandem in space, we're talking about beam type detectors now. And so we use these type detectors to create highly reproducible quantitative analyses. And then we use these as triple quadrupoles, where you have 3 separate quadrupoles. The first quadrupole separates all the peptide ions from each other. The second quadrupole fragments those, and the third quadrupole reads out all of those fragments. And it's how you program that instrument up to give you targeted proteomics. And then more lately, we've got the time of flight instrument, which is an instrument now that replaces that third quadrupole with a time of flight to get back to high resolution and high mass accuracy mass spectrometry. And this has been a favor now for this data independent or swath analysis, which we'll teach you about. And so this is really the process of that type of instrument. You have still your first quadrupole, which you separate out your peptides in the first instance. You fragment them in the collision cell, and then it enters into an evacuated tube, and they get pushed down this actual space of this tube, and they get turned around by a deflector here to actually get twice the flight path length, to give you twice the resolution and get a tighter mass accuracy. And these instruments are sold by companies like SciEx, the Triple TOF, or 5600 or 6600 you might know about, and they also sell the nanospray LCs that go on the front of those, and that's the ones that we look for. But when we talk about data identification, we always talk about, well, what's the modern aspect of all of this? But we really class that as not being really modern, which is really current day, because the tools need to continually be updated and also improved as we go through. And so we're going to teach you where we are with the current day tools, give you a bit of an insight into what we're doing in the future. But you can already see that with these tools that we have, there's still a lot of work that needs to be done, and trying to understand that is where we want to get you to a point where you can understand really what's going on. So with data collection and analysis, this is the very simplistic view of what's happened. So we have a mass spectrometer that if you put ions into there, you have an analyzer and a detector, which will give you a spectrum, which really just tells you what the peptide mass is of all of those components going into the mass spectrometer. If you took just those peptide masses and you knew what the sequence of your protein was, you could calculate out what those individual peptides if this was a tryptic digest, what was the mass of each of these individual tryptic peptides, and you could work out from that as a peptide spectrum or a peptide match spectrum where you're just matching each of the individual peptides. Problem is, we don't have the resolution, we don't have the mass accuracy enough, because there are peptides that are also isobaric, and even though they have a different structure, they have exactly the same mass, and so it doesn't allow you to distinguish those peptides from real ones from the ones that you actually have. So this is a tool that you can do fingerprinting, but you still need fragment data, and so this is what we call tandem mass spectrometry. And so if you fragment a peptide in the mass spectrometer, that's called tandem because you take the first component, the mass of the peptide, then you fragment it to get the masses of all of that parent. And this is what you get here. And so if you add a third quadrupole or a time of flight tube at the back end of a mass spectrometer or an Orbitrap, you can trap your peptide in the first instance here, fragment it, and read out all the fragments. And now you're getting the fragment profile of that particular peptide. And then you can read off what that sequence is by just looking at the mass difference between each of those peptide fragments were. And so that's what we do. And so generally, this is what happens in a mass spectrometer. We have a peptide. It has a neutral mass that we can calculate from each of the residue masses of each of the amino acids. And if we add on protons, and we'll have a proton added onto the C terminus and also the N terminus, and so it'll give us what we call a doubly charged ion. That doubly charged ion in the mass spectrometer then it has its molecular weight divided by 2 because it's a mass over charge measurement. And then each of those fragments then come back as a single charge mass of each of those. And it's those fragments which we need to understand. And it's not as if you just get a single ladder, you're getting a ladder from both the N terminus and also the C terminus, and so they overlap each other. But if you had a pure spectrum, you can calculate what that would really be. And we'll teach you how to do that this week as well. And so in the end, really, for proteomics, data interrogation is really the Achilles heel. If you don't have the tools available to you, and if you don't have the expertise in understanding those tools, it's very difficult to move forward from a pretty standard list. And you need to understand the list that you have, because there's data in your analyses that you may not be aware of that you've missed out on, or there's an over-interpretation of what you're doing. And so you need to understand what those different nuances are to make sure that your data set is actually correct. And this is describing just the basics of what we're doing. So if you start off with a protein sample, each of those proteins can be divided into a number of different peptides. Problem is that there could be also shared peptides that have exactly the same sequence between separate different proteins. And so now, by dividing out your protein into peptides, you've lost that connection from the peptide back to the protein. And so when you try and make that identification, you could be misidentifying that protein that you're after. But the mass spectrometer really only wants to see peptides because of its mass range. And you look at, when you're doing your fragmentation, you're getting each of those peptides being fragmented. Then you do what we do at this point here is the database searching. And that's what we're going to be teaching you about this week, and where the TPP comes in the trans-proteomic pipeline. So once you do that peptide identification, you may be able to identify all of the peptides, or you may miss out on some of those peptides because they may not have fragments that are suitable for identification. And so at this point, you can already see that a couple of peptides will not be able to be recovered. And so when you're trying to identify all of these proteins, this one here, protein A, that had 2 peptides, is now missing 1 peptide and only has 1 peptide left to do its identification. There are proteins in, say, the human proteome that you cannot detect because you don't have peptides for them. And so this is the typical situation here. If we weren't able to recover that peptide, we'd lose that identification. But luckily enough, we have still 1 peptide. It's unique to that protein, so we can identify that. But there are other situations where you could have 2 peptides that are unique to that protein. Then a third peptide could also be identified with that protein, but it also could identify another protein. And so now you have this problem of, did I identify the protein correctly, or did I identify the other protein correctly? And so this issue of protein inference comes a lot into play, and we'll talk to you about that when we talk about the protein profit area. So this is what we have at the moment. This is the Trans-proteomic pipeline, all of different tools. So it's really a suite of many separate tools that are interconnected, and not all the tools you want to use all at the same time. There's some tools that you want to use at different points of the time to end up with a list of proteins that has not only correct identification, but also statistically valid as well. So we'll talk to you about how we take the raw data all the way through the data formats that we have, the different types of search engines that we use, the different types of search engines that we use. We have the tools, statistical validation tools, both at the peptide level, the PTM level as well for post-translational modifications, and trying to statistically define exactly where that post-translational modification is, looking at quantitation tools, and also protein profit. And beyond that, then we take those identifications and looking at the different resources that we have. So in that respect, we have a number of different aspects that we'll be teaching you about there. And then finally, we have new tools which we call Respect, which will also teach you how to actually reanalyze your data, because you may have what we call chimeric spectra. And this is spectra which you'll have two peptides being fragmented at the same time, but the search engine only identifies one, but leaves the other one behind. And so now we're getting to this point of where we can take that spectrum, reanalyze that, and bring back that second identification, or third, or fourth, depending on how many peptides were actually captured at that same time. And this is a tool we call Respect. We have our what we call our peptide compendiums, the peptide atlas. And so once you get beyond identifying all your mass spectrometry based identification of peptides, how you can utilize other resources that are available on the web, such as your PRIDE, which is the primary resource at the moment, through Proteome Exchange, which is a real boon for raw data repositories, and so you can reanalyze other people's data, and then take them through the TPP. And what we do is we take all of this public information that we can get hold of, and each year we analyze the human proteome based on all of the data that has been produced within that year, plus all the previous data. We put that all together, reanalyze all the data with our new improved tools that we keep on going through, and then deposit this is what we call the peptide atlas. So each year we've produced a status paper, the status of the human proteome, and we correct for misidentifications that people have made in the past, and we serve that all back to the community, and we'll teach you how to utilize those tools to the best for your experiments. And finally, we'll talk about some quantitative proteomics, not only at the shotgun mass spectrometry level, but also data independent and also SRM targeted mass spectrometry. But it's particularly well suited for doing a lot of identifications at the quantitative range for biological questions, looking at protein level changes, how we can utilize those, and then also the tools that we have for that analysis and part of the Trans-proteomic pipeline. This is just an example of one of the areas, and so if you're doing a different growth curve of two different cell lines, you can do one with, you can grow one cell line with a mixture of modified amino acids in there, the lysine and arginine, and so the cells will uptake those amino acids as the proteins are being expressed, and so all of your proteins will now have this modification on there, and then you take your control cell line, then you can mix those two together, do your mass spectrometry analysis, and then look at these differences just based on a mass shift. And this is where you're looking at what we call either SILAC type experiments, and if you don't have that capability of doing this type of growth curve or growth analysis, you could do this as a labeled or chemical labeling approach and do it that way as well, and also achieve a very similar type of result. But in the end, what you get is prototypic peptides, and we want to understand which are the best peptides to use. And so understanding your lists and working out what peptides make it best to uniquely identify a protein, so you can do these targeted type quantitative experiments in the end as well. And these prototypic peptides are what we class as not only unique to the protein, but also likely observed. And so this is one aspect that people need to understand as well, that not all peptides can be observed in the mass spectrometer. And if you're trying to use those peptides as well, not all peptides can be used as standards either. And so that really cuts down your list of what you'll be able to use as we go forward. And so proteotypic peptides are useful for when you're trying to do discovery experiments in quantitative proteomics. They're best for creating these targeted lists in SRM transitions, and also they're useful to make these heavy labeled versions of those, so you can be used as a quantitative standard because you can measure them specifically. Finally, some of the tools that we'll talk about is not only the targeted proteomics or the selected reaction monitoring and the resources we have available as part of the SRM Atlas. And this is where we utilize the mass spectrometer by now selecting only or hopefully a single peptide in the first quadrupole, rejecting all other peptides, fragmenting that particular peptide, and then looking at all of the different fragment one by one to get these quantitative peaks. And so what we'll teach you is the resources that we have that you can, without having to do all of this experimentation up front, you can take these resources down and start to measure straight away. We'll also teach you the tools that we have for the swath analysis or the data-independent analysis, which is expanded on this whole process, where we now take multiple peptides in the first quadrupole, fragment them all at the same time, and read out all of those different fragments. And so you now get a very convoluted list of peptide fragments being detected at the same time, and the tools that we have that enable you to analyze those, not only in terms of detecting them for quantitation, but also doing statistical validation, and also, more recently, modification analysis of those as well as part of our swath profit PTM. And so, just to go back to what the swath really is, again, this is a technique where the mass spectrometer is not told to look at a particular peptide, it's told to look at mass ranges. And so any peptide that fits in those mass ranges will be fragmented at the same time. And this happens in a series in which all of the mass ranges are performed in a particular sweep and then started again. So this usually takes about 2.5 to 3 seconds to perform. And so you get all of those peptides being fragmented at once. But you need the tools to be able to analyze all of those at the end. And so finally, what we're going to do is have 5 days worth of fun. It's not going to be just sitting here and just being spoon-fed. You're going to be requested to do the analysis yourself. The programs will be on your computer hopefully by now. If they're not, please tell us. And then, in the background, Louise and David and Mike, Sam, and Uli can walk around and help you with your computers at this point. And we'll go through each of the different aspects of fundamentals in terms of what we're actually doing, and then making sure that you can actually do those analyses yourself. So hopefully by the end of the 5 days, you'll be independent enough so that you'll be asking the same questions that we ask, and then you'll be starting to challenge us. Why can't we do this with this? Why can't you do that with that? And then we'll be also asking you for, because all of our tools are open source and freely available, that maybe you want to do some programming yourself and contribute some tools there as well. So we see this as a two-way street. So hopefully you'll come away from all of this now, get all of this information into your own experiments, and get to understand how to do that. And so we expect this to really be 5 days worth of fun. And so we'll leave that at this point, and we'll talk about when we start off in the session straight after tea. Thank you. All right, so we might as well start the course. So as you can see, this is the structure of what we're going to have in the course. We'll have each of the modules broken out into separate components. And so they'll take you through the very initial stages of database searching, the data formats, then getting into some of the tools in terms of statistical validation, which will just start on towards the end of the day. And then we'll continue that on tomorrow, and then how you can get into much larger searching into cloud-based components with Amazon Cloud, and then getting into the quantitation tools, and that leads off into our other aspects of cross-linking, the whole proteome resources of Peptide Atlas, SRM Atlas, and swath analysis. And then the last 2 days will be our efforts in giving you tutorials and exercises, so that all of the things that you've learnt within the first couple of days you'll start to be taking them through those exercises, analyzing those pieces of data sets as we do that together on the screen, and see that you're going to get the same performance. So we'll be utilizing also the internet, and so we'll be going back into Seattle, analyzing some of the results that we're pinging the servers back there, and then you'll get those results here at the same time. So that's the process at which we'll take. So I'll be just introducing each of the people as they come through, so you get a bit of a sense of who they are and what they're actually doing. But then just give you just a brief background on a lot of that stuff. So Mike Hoopman will be starting us off at the moment. So Mike has quite an extensive experience and expertise in database search algorithms. He's a primary author of Comet, which is the open source version of Sequest. Now, Sequest, you know, was developed just over 20 years ago by John Yates and Jimmy Yang. John was the brains behind all of the analysis of peptides and their spectrums when he was doing this in Don Hunt's group, and together with Jimmy put together a computational program to push that forward so people could automatically analyze their peptides. Now, going on beyond that, Sequest has been available for many years. It's been also commercially available, so if you've used any of the Thermo software, you've probably used Sequest. But as that hasn't been updated in many years, finally the decision was made by the University of Washington to release that software as an open source. And so Mike and Jimmy got together and wrote the Comet version, which you can now access. And we have that all included in the TPP. So Mike will be talking about that. So in terms of expertise and knowledge, you couldn't get anybody closer than that. So this is where it fits in. We'll talk about the database searching at this component. There is a smaller component that we'll talk about in the next module, which is data formats, but that also takes into data formats that it comes out of these algorithms as well. Because you remember, these algorithms are all written at different times, different points, and so different formats come across. And so one of the big boons of the TPP, the Trans-proteomic pipeline, was to try and bring a common format through all of these. And even though at the start of the Trans-proteomic pipeline a common format was developed, was also improved beyond that. And so the latest data formats we'll be talking about and how they interact with the TPP. So as we go through this whole program, you'll go through getting your raw data from any different mass spectrometer, going through either open source, free type search algorithms or even commercial search algorithms such as Mascot, and then being able to take that data back into the TPP and do all your analysis. Again, as I said previously, we also have Respect. And that's going to be talked about as well, Respect? Yes. Yes, okay. So there'll be that tool, which is a unique tool that enables you to analyze these chimeric spectra and really get these other peptides that you're not normally seeing to be also identified using this software. And so it's almost like a leave no peptide behind approach. And so the TPP tools is what we have as a set of tools that will come through from database searching, which is your primary. We usually use Comet as our first search algorithms. Each of the search algorithms are slightly different from each other, so combining them gives you an advantage as well. And we'll talk about that combining too. And then that fits into areas where you then validate and then use programs like iProfit, which is really a word for Interprofit, to take your results, combine them together, and normalize that score across all of the different search algorithms. And so not every search algorithm is written the same way, and so they all come across with different scores. And so how do you combine that? And so these are the modules that Mike will talk about now. Then as we go towards the end of the day, we'll just start to touch on the peptide profit and how you can make these statistical validations on those results, and then also start to create spectral libraries as well. And then the quantitation will be led off on tomorrow. So with that, I'm going to finish and allow the expert to talk. And at any time, if you want to stop him, feel free to ask a question, and Mike will most certainly answer that for you.
All right. Thank you, Rob. Hi, everyone. Good morning. As Rob had introduced, I'm going to be talking about tandem spectrum database searching in a very general sense at this point in the course. And then as the course progresses, you'll start actually using a lot of search engines and get a more in-depth knowledge of how they work and the various ways you can use them. But as just kind of our overview of what I will cover, there will be some basic background, and this includes the generation of spectra as well, and their fragmentation in the mass spectrometer to generate tandem mass spectra, which are searched in these algorithms, as well as actually showing you examples of those spectra and how one would interpret them in the absence of an algorithm. And the best way to understand the algorithm is to first step aside and see how would you solve this problem yourself. And the algorithm is really just the automation of that process. And then finally, the algorithm itself, how it interprets spectra and how those spectra are validated. Rob had shown this picture previously of our entire trans-proteomic pipeline, starting from raw mass spec data here at the left side, working its way through to eventually identifying proteins. This particular portion of the lecture is just this peptide identification part seen here. And there are several algorithms that can be used for peptide identification, of which, of the 4 listed here, this is certainly not all the ones possible, but of the 4 listed here, only the ones kind of highlighted in darker colors are relevant to this portion. These are database search algorithms, with the exception of Spectrast, which is a spectral library matching algorithm, which is in a different lecture. One important distinction, I guess, to make in all of this is that despite there being many different algorithms, they can be plugged into the trans-proteomic pipeline, or plugged out of it, and used in kind of a cassette fashion. And so some of these algorithms that are listed up here have this asterisk indicating that they are commercial software, not part of the TPP. You might have them in your labs or on computers that you work with, and if you do, you can still use the TPP with that software. If you do not have any of that commercial software, the TPP does come with free software, quite specifically here, XTandem, as well as other algorithms such as Comet, which can be downloaded off the web and then plugged into the TPP. So I'm going to start at the beginning of shotgun proteomics, and I'm going to use a lot of terms interchangeably for shotgun. I'm going to use tandem mass spectrometry to sometimes indicate shotgun proteomics. What is meant here with shotgun proteomics is that you start with a sample of proteins, but you don't know what those proteins are. And the way that you solve this is to first break them into smaller pieces called peptides and then analyze them in a mass spectrometer in kind of a semi-random fashion. That we typically call data-dependent acquisition, and thus that's kind of semi-randomization. First fragmenting all of the proteins into peptides and then kind of randomly analyzing them is where the term shotgun comes from. It's actually a borrowed term from the field of genomics, where they had originally analyzed and sequenced a lot of genomes in much the same fashion, break chromosomes into small parts and then analyze them individually. And following that analogy, the way genomes were assembled is all of those pieces were then reassembled into contigs and then from there assembled back into chromosomes. So likewise with proteomics, we take the spectra that we've acquired on the mass spectrometer and we interpret those into the peptide sequences that derive those spectra. And then from there we can infer what proteins were in our sample. So this is pretty much the cycle here, starting from proteins, working our way down to data, and then performing all the analysis back to proteins. So one of the terms here that you will see a lot is peptide spectrum matching. So protein identification relies on peptide spectrum matching, and that's essentially assigning a peptide sequence to each of these spectra. And so I often refer to these as PSMs, and you will see that term probably throughout the remaining 5 days of the course. So at the very basic level, we're going to graphically represent this whole process from protein down to spectra. We start here on the left with our protein that through a process, a digestion process, which can be chemical or enzymatic, but typically it's enzymatic, we break our proteins down into smaller pieces of peptides. Those peptides are then ionized through a process usually called electrospray, which is coupled to chromatography as a way to separate and simplify our mixture of peptides. And then once ionized, they can be analyzed inside the mass spectrometer. And so what you have from this is a primary spectrum, often referred to just as an MS, mass spectrum here, where each of these peptides, which each have their own different characteristics, are represented by a different peak. And those peaks are related to the mass and the number of protons that the peptide carries. And so larger peptides will have more mass and will shift towards the right end of the spectrum, but as they continue to acquire more protons, you divide by that number of protons, and that will bring them back to the left. And so in this way, we can analyze each peptide by its mass in the mass analyzer. But for the purposes of identification, we need to take this process a step further. And so what usually happens then, this is what we saw before, protein to peptides, to analysis in the mass spectrometer to produce this MS spectrum. What we usually do at this point is we isolate one of these peptides of interest here so that we can then fragment it by itself inside the mass analyzer and analyze each of those fragments in what's known as a tandem mass spectrum, or MSMS, or you might even hear me say MS2 in some cases. These are all various terms that have been used throughout the literature, and they're interchangeable. And what this does is give us a barcode, for example, representing the different amino acid compositions of that peptide, specifically that peptide that was isolated. And it is this MS spectrum here that the database search algorithms interpret to identify our peptides. So this is a very important step in this whole analysis process, and I will take you through the details of this spectrum. But first, I'll give you a little bit more information on how the data is acquired in the mass spectrometer and the differences between the MS and tandem spectra. The mass spectrometer is usually operated in this alternating mode where peptides are analyzed first intact in these MS spectra, labeled here with the blue arrows. And then in between each one of these analyses, we isolate and fragment different peptides from that original precursor MS analysis to get all of our MS events. And this happens in a continuous fashion over the entire sample analysis, typically 1 to 2 hours of liquid chromatography. And it's done in this fashion because these peptides here, these precursor ions here, are going to change over time as your chromatography conditions change and different peptides elute off of your column. And so over here in the upper right is really just a two-dimensional representation of just that, where all of the different peptides that were detectable in the sample are shown here with their mass over charge values on the y-axis, and then the time at which they eluted off of the chromatography column and were analyzed by the mass spectrometer on the x-axis. And so if we look at this mass versus intensity versus time, our three-dimensional graph here, we can see for any given peptide that there is a point at which it starts coming off of the column, point at which it reaches its maximum intensity off of the column, and then finally where it decreases and it's no longer seen. And along each of these MS scans here, the profile actually changes where, given enough time, these peptides would no longer be observed to be replaced by other peptides that are being analyzed. The peptides themselves, of course, are comprised of amino acids, and so when we measure the mass of peptides, we are quite specifically measuring amino acid masses and then eventually, of course, the atomic masses inside each of those amino acids. But it's important to note that the 20 amino acids all have different masses, with the exception of leucine and isoleucine, which have identical atomic composition and mass. But it's this key feature right here, this difference in mass for each of these amino acids, that allows us to sequence peptides. Before I get into the details of that, what you will see when looking at mass representations of peptides and proteins are the difference between the monoisotopic and the average mass. So any peptide observed in a mass spectrometer is actually represented as a series of isotope peaks, in that each of the atoms that comprise peptides have heavy stable isotopes in them. Those heavy stable isotopes create a mass shift. And so what you usually see, that's the reason for seeing this isotope distribution, in that if you happen to have a molecule that contains none of those heavy isotopes, you get what's known as this monoisotopic peak. And for most peptides, it'll be a tall first peak here. But then for some small fraction of the molecules of that peptide, there will be one or more heavy stable isotopes, creating this mass shift. And so you get a smaller representation of molecules that are heavier by 1 Dalton, and then others heavier by 2, and still others even 3 Daltons. Among this isotope envelope here, you can determine the charge state of your peptide, or the number of protons currently residing on that peptide, by the difference in mass between any two peaks in the isotope distribution. And the charge state is simply the 1 over the difference in that mass. And then the other way that peptides are sometimes represented is this average mass. And average mass is, as it says here, the centroid of the isotopic envelope. So if you were to represent all of the average of all of these peak areas together in a single value, that would be the average mass. So it's this monoisotopic peak then averaged by the contributions of all these other isotope peaks. There's an example over here for this peptide that begins DIG. There is a monoisotopic mass, and then its average mass is slightly higher by a little more than a Dalton. The larger your peptide, the larger this average mass is going to be. Many years ago, when shotgun mass spectrometry was done almost exclusively at low resolution, these isotope peaks could not be resolved, and algorithms and the people analyzing the data relied heavily on this average mass. These days, mass spectrometers typically acquire much higher resolution, if not for the tandem mass spectra, at least for the precursor mass spectra. And so most of the algorithms now have been adapted to use almost exclusively this monoisotopic mass. So tandem mass spectra are comprised of fragment ions. And to give you an example of where these fragment ions come from, I have here a short 4 amino acid peptide sequence with each amino acid just represented by an R group here. And what happens is when energy is applied to the peptide for fragmentation, that it has usually consistent breakpoints. And when using CID or HCD, so collision-induced dissociation, or higher energy collision-induced dissociation, These breakpoints tend to be between this carbon and this nitrogen atom, between each of the amino acids right here. And what you get are what's known as B and Y ions. So the B ions, everything on the N-terminal side of this breakage, and the Y ions, everything on the C-terminal side of the breakage. And in this 4 amino acid stretch here, there are 3 possible B and Y ions. They are numbered from lowest mass to highest mass. So when reading the peptide from N to C terminus, the B ions are numbered 1, 2, and 3. But when you number the Y ions, they're numbered from the C terminus to the N terminus. These aren't the only possible breakages that could occur. If you were to break on the other side of this carboxyl group here, we have what's known as A and X ions. X ions are hardly ever seen in the mass spectrometer with CID fragmentation, but you do occasionally get these low mass A ions, in particular the A2 ion. And then finally, if you have breakage on the other side of this amine here, you get C and Z ions. And C and Z ions are typically seen in ETD type dissociations, but on very rare occasion can be seen in CID. There's also other obscure fragment ion types. I'm not going to go into the details. I don't actually ever look for these, nor do I recall recently ever having found one of significance in any spectrum I've looked for. But these are just other obscure breakages that can occur. And just realize if you ever see these in the literature that there are these other definitions for ions, and that this is the reason for these definitions. And then other ions that might be observed in a mass spectrum are ammonium ions. And ammonium ions are essentially, they're a double breakpoint that isolates one of the amino acids from the rest of the peptide. And there is some bias that certain amino acids tend to show up as ammonium ions more than others, such as proline or valine. So absent in this for whatever reason, or not for whatever reason, but typically absent in this breakage here is this lysine and arginine, because this represents what's usually seen amongst tryptic peptides, and this bias comes from the fact that the lysine and arginine are almost exclusively on the C-terminus of every peptide, and so might not be exposed to this same fragmentation frequency as amino acids interspersed throughout the peptide. So it's important to understand where these ammonium ions come from, because they actually can be used as a diagnostic in interpreting a mass spectrum. To give an example here, here's a MALDI-TOF-TOF tandem spectrum. And in the low mass region, you can look through here and see 3 ammonium ions, a proline, isoleucine, or a leucine, we can't tell them apart, or phenylalanine. And what this gives us is this short little, I don't want to say a tag because they're not necessarily consecutively related, but 3 amino acids that should be present in the peptide sequence that is represented by this spectrum. And so if this spectrum were solved, and we're going to jump to the end and solve it for you, you get this peptide here, this rabbit glycogen phosphorylase, and we see that it does indeed have a proline, a phenylalanine, and a leucine in it. So this initial ammonium ion evidence helped us to identify this peptide sequence. The remaining fragment ion peaks here, you can see are B and Y ions and the A2 ion seen over here. So that brings me into peptide fragmentation, or where each of these peaks comes from and how we determined that each of these fragment ions was related specifically to this sequence. Well, if we take our actual peptide sequence here and we create fragmentation to produce our B and Y ions, as described earlier, we see that it's a an 8 amino acid sequence here, so that there should be 7 B and Y ions produced, and they're produced specifically breaking between each amino acid here, where the B ions, the mass of each B ion as we move down the list is the sum of the amino acids masses plus a proton, and then the Y ion masses here are the sums of the amino acids on the C terminus of each break point plus water and a proton. And so this list of masses is now what we would expect to observe in our mass spectrum. So we can take our spectrum again, as before, and assign all of our B ions and step our way along from N to C, because this is the B ion series, step our way along and label each of the peaks that a B ion matches. Now, occasionally one is missed because of some sort of fragmentation bias that might occur in the mass spectrometer or an interfering signal. So it's not always going to be perfect, but in a good scenario with a good spectrum, we do, in fact, explain a lot of these tall peaks by the B ion series. We can then repeat the same process, this time from the C terminus to the N terminus. So it's the same peptide, but now the letters are in reverse here, and label all of our Y ions. And so putting that together, we can now label every major peak with B and Y ions. And then additionally, we can go through and look at all of these smaller peaks and try to explain them away as, in this case, an A ion. Here's a Z ion that happened to appear, another A ion, and so forth, and try to interpret as many of the observed peaks as possible. And this is essentially how a spectrum is solved using a fragmentation analysis. Now, algorithmically, when you do this by hand, it's tedious. It takes a long time. You don't want to do this for the tens of thousands of spectra you can generate in a single hour. So algorithmically, several approaches have been developed over the past 2 decades. One of them is de novo sequencing, which is essentially just taking what you would do manually and applying a computer algorithm to it. So de novo sequencing starts at any given peak in an analysis and then computes the difference in mass between each peaks and tries to step its way all the way to the other end of the spectrum to produce a peptide sequence. It then hopes, once it hits the other end of the spectrum, to step along other unexplained peaks all the way to the right and produce the same peptide sequence. There are several tools for this. Some of the more famous ones are probably PepNova over here and Peaks, and maybe even Lutefisk that you've heard of. Peaks, by far, I think, is the most renowned algorithm for this. They work well for small peptides and for for very clean, easily to interpret spectra, but they don't work very well for all spectra. And as such, usually de novo sequencing is augmented with other types of algorithms. And that's where the TPP comes in, where TPP uses these uninterpreted MSMS database search algorithms. And so these take a different approach on sequencing, and the bulk of the remaining part of this lecture is going to be on how these algorithms work. In a nutshell, you just hand the algorithm your entire set of uninterpreted tandem mass spectra and a protein sequence database. And you tell this algorithm to find peptides within this protein sequence database that can explain these spectra. And it's not going to do this analysis in a de novo way. There's actually many different types of scoring algorithms and approaches that are used, and I'll go over some of those. But first, the basic input is your fragmentation spectra. It helps to have your precursor mass, the mass that was isolated, but if you can have this down not just to the mass that was isolated, but specifically the monoisotopic peak of the peptide that was isolated, that is the best information possible, and the charge state, and of course your sequence database. These steps, there's only 5 of them involved in database search algorithms. One is to identify peptides that equal the precursor mass of any of the spectra being analyzed, theoretically fragment the peptides, or create a theoretical spectrum that you think might actually look like the real spectrum, compare that theoretical spectrum to the actual one, generate a score, and then rank all of those scores to find the best matches that explain the spectra. An example of this in small scale here is if we have our spectrum on the left and our database sequence on the right. We know the precursor mass of this peptide. Inside this sequence database, there are 3 possible peptides that all have the same precursor mass, and so they're all candidates for creating this spectrum. You can then create a theoretical fragmentation set for each of these peptides and compare that to the actual observed spectra. One of those peptides, in this case, matches very well, has a very high similarity score. The other peptides are not, the fragments that were generated for these guys don't resemble this spectrum at all. And so among these, you would then conclude that this first peptide is the best match for the spectrum you are looking at. So there are many parameters that go into the search algorithm, in addition to the very low number of actual, the low number of data inputs. But there are actually a lot of search parameters. And the first is the actual choosing the correct database. And search algorithms can actually take a variety of databases. And you can develop, even in some cases, I know Sequest was capable of doing this, creating sequence databases from raw genomic data and just going through this and interpreting amino acids from the raw genomic data to find strings of amino acids that might belong to proteins and might therefore be represented by your spectra. This is probably the most difficult approach to start from. So better yet would be to use transcripts or ESTs, which are genetic data that hopefully represents just actual proteins being produced by a cell. And then finally, actual protein libraries, which are perhaps the easiest to work with, which are actual protein, the genetic information has already been translated into protein sequence, and these would be the actual proteins you are hunting for in your sample. Optionally, you can include enzyme specificity in the search, and this is important from a computational point of view. If you were to take some protein sequence, as seen here, and compute the number of peptides produced from that protein sequence, if you put an enzymatic constraint on there of, say, trypsin, then all of your peptides, expected peptides, would be bounded on the right side by a lysine or an arginine, the actual cut site. And then the next peptide would start from the following amino acid. And so the number of peptides you could observe from this protein is actually very small relative to all possible peptide combinations in a protein. To give a better example of this, if you consider an unconstrained, enzyme unconstrained number of peptides, for a given protein sequence, there is an enormous number of peptides that are possible. I'm only representing a few here with these blue lines, but essentially you can start from any amino acid and end at any other amino acid and have any length along the way in between, which creates a very large subset of peptides for that protein. With the tryptic enzyme, however, you are limited to just these amino acids here if you had complete tryptic digestion. So the number of possible peptides you need to search for is very small. And computationally, this is actually quite important. So if I expand this example here to include an entire human proteome, and this is the human IPI database, so it has proteins plus protein isoforms and nearly 50,000 entries. If we were to consider a single peptide of mass 1,000 Daltons, the number of tryptic peptides in the whole proteome is only a little more than 1,400. But the number of unconstrained peptides in the whole proteome is over 300,000, which is a 225-fold increase in eventually what will be computation in order to search this entire proteome against a single spectrum with a precursor mass of 1,000 Daltons. It gets worse as you get into larger and larger masses. If your peptide is larger in mass, in terms of tryptic peptides possible, there's a smaller number, but the number of unconstrained peptides is really just as large. So your computation can be over 1,000-fold more using an unconstrained search. Additional parameters in your search database are post-translational modifications that you might want to identify. These are very important in that they often represent some additional functionality or changes in functionality of proteins and their interactions with other proteins. So they're of huge biological importance and also somewhat of a headache for the computational guys that need to solve post-translational modifications in their algorithms. There are different types of PTMs. The first is static modifications, which are modifications that occur on all occurrences of an amino acid, and these are typically the result of some sort of sample preparation. For example, when we process proteins, we usually break disulfide bonds and then alkylate those bonds to prevent them from reforming. That alkylation occurs on cysteines and creates a modification that is then applied to all cysteines and will be there in the analysis downstream. Static modifications are computationally very easy to deal with. Variable modifications, on the other hand, may or may not be present on a peptide, and they may or may not be present in different combinations. And so that combinatorial problem creates a lot of additional computation. And should also point out that modifications can be on any residue, depending on the type of modification, or even on the termini of the peptides. And those termini can be actually naturally occurring, such as the case of the N-terminal peptide or C-terminal peptide of a protein, or as a result of some sort of experimental design, such as SILAC labeling, where after tryptic digestion, the label is usually contained on the C-terminal amino acid. So to give an example of variable modifications, we have, again, our peptide that starts with DIGS. And if we were to consider serine phosphorylation on this peptide, instead of just searching it once, we now have to search it 4 times for each of the possible places where a serine can be phosphorylated. But as you know, phosphorylation can also occur on threonine or possibly even tyrosine. And so if we were to consider threonines and tyrosines as well, all possible combinations of phosphorylation, actually the number of possible forms for the single peptide that you need to consider is now 16. So it actually grows exponentially with the number of possible modifications that reside on your peptide. And if we consider this, again, in the context of the entire search, we go back and we look at our single spectrum with a precursor mass of 1,000 Daltons and the number of tryptic peptides we have, well, if we consider phosphorylation on that, then we actually increase our computation 3.5-fold for just that single spectrum. If we use a different spectrum with a larger precursor mass, sure, there's fewer peptides, but they're larger peptides, and so they have more potential phosphorylation sites. The number of possible combinations explodes to almost 17,000, so it's a 67-fold increase. If we can then go even further and say, well, what if we don't have tryptic specificity in our analysis and we consider the unconstrained peptides? These were the number of searches that need to be performed on an unconstrained level. If you add phosphorylation to that, it jumps quite quickly into the millions of possible combinations. And this is computationally horrendous here. Have to consider 15 million different combinations of peptides plus their phosphorylation where the much smaller problem here would be almost 17,000 for just phosphorylation, then just trypsin, 250. So these are all considerations that need to be made when setting up and performing your database search. Typically, you don't search for any sort of post-translational modifications that, one, you do not expect to see, or two, you are not interested in finding. And that keeps the computation down to a reasonable level. You also need to provide the correct fragmentation types. As I alluded to earlier, CID and HCD methods of fragmentation produce primarily B and Y ions. It says here most search engines will only match against these types. This is perhaps a tad out of date in that a lot of them now will incorporate ETD spectra. But this is true that nearly every search algorithm out there, B and Y ions are the default standard. You don't, when looking at CID and HCD fragments, you are not interested in matching C and Z ions. Adding those extra ions just increases a lot of computation. It also increases a lot of the noise, which will add confusion to the algorithm, and performance can drop off as a result of spending additional time and resources looking for things you will not find. ETD, on the other hand, mostly generates C and Z ions, and so algorithms that work for both CID and ETD usually have some sort of toggle in the parameters. If you use ETD, then switch over to analyzing C and Z ions. Some mass spectra have mixed mode, CID plus ETD or HCD plus ETD, and in those cases you might actually want to use all four BY, C, and Z ions. It's good to know that QTOF instruments can produce A ions, and so you might want to supplement A ions if you're using a QTOF. And then finally, the search software itself that you use can have a huge influence on the results that you obtain. And so I'll go over some of the algorithms that are out there, some of the major ones that are out there. Certainly, this is not exhaustive. I think 10 years ago, there was some 30 primary search algorithms. That was 10 years ago. These days, I would say 30 is probably the most anybody who is really knowledgeable can identify, but there's probably 50, 60, 70. Maybe we're up to 100 now. You really have to dig through the literature to find them all. Sequest, though, is kind of the parent to all of those algorithms. This was the one that showed the world how easy it was to create this uninterpreted tandem mass spectra algorithm search program that could take databases, entire sequence databases, and entire sets of spectra and produce meaningful results. It is actually still used today, so this is how good of an algorithm it is. It's survived more than 2 decades of further developments in the field. It uses, the original Sequest algorithm used this 2-step approach to break computation down. The first was to develop this preliminary score, SP score, and then finally to do a cross-correlation amongst the best preliminary scores. This cross-correlation, it's a dot product with correlation, was ultimately the scoring metric for results from Sequest. Since this has actually only just recently become free due to a restrictive license when it was first released. That license was needed to be purchased directly from the University of Washington or from Thermo, who had exclusive rights to the license. And so it was one of the limitations in its availability. But today, there's other options. Comet, once the patent expired on Sequest, Jimmy Ang spearheaded the effort to rewrite the algorithm as Comet, which is the modern-day version of Sequest, but the scoring algorithm is essentially the same. This is my favorite search engine. As Rob might have mentioned, I am one of the developers on it. So I might be a little biased, but I really think it's actually a wonderful search engine. My interest actually, I was a student at UW, and so I actually was able to work on the Sequest code when it was private years ago, and that's how I came to understand how search engines work, and that's how I was able to contribute to the development of the modernized version. Other efforts out there, XTandem is a separate search engine, but it has a feature called pluggable scoring. I think it was actually developed by Brendan, who's teaching the other Skyline course right now. Developed a pluggable scoring for Tandem, and this pluggable scoring algorithm known as the K-score replicated the Sequest algorithm inside Tandem to some degree. And so this is also an alternative if you want a Sequest-like search. The actual cross-correlation algorithm is shown here. I won't go into too much of the details because it is a lot of math to explain it start to finish. But essentially, the cross-correlation looks at the best matching given a series of offsets to align a theoretical fragment spectrum to an actual spectrum. And then the maximum here is the actual score that is taken for the cross-correlation score. An alternative, also commercially, but perhaps one of the most widely used search engines in the world is the Mascot search engine. So many of you might have actually seen this before or have a copy in your lab. It uses a probabilistic scoring algorithm. I can't actually tell you the details because it's black box. It's still privately held, and so we don't know how it works, but it's really simple to use, and so that's why it's so ubiquitous in the field. And so you might actually end up using this a lot in your own research, and this slide is here to let you know that you can take results from this algorithm, even though it's commercial, and plug it into the TPP and continue doing all of your data analysis within the TPP. So if you are down one of these commercial avenues here, it is not a problem. XTandem is another open source algorithm developed by Ron Beavis. It was actually, when it was developed, it was very, very fast, much faster than Sequest, which made it very useful for people for getting through large data sets. Its scoring algorithm, like Sequest, it uses a dot product, but it creates this hypergeometric distribution, which incorporates this factorial, bonus on top of the score based on the number of B and Y ions in a match. So it's not merely matching ion peaks and their intensities, which the dot product contributes to, but also the frequency at which you identify those peaks contributes to the score. It has a web interface and also a command line interface, and this algorithm is included in the TPP, and so you will actually be using this as part of the tutorials in the course later in about day 2, 3, 4, somewhere around there. XTandem also has a feature known as refinement mode that you may or may not see in other algorithms. And in this, it's a multi-pass way of searching, where you make your first pass against your full database and a limited number of parameters, such as search just for tryptic peptides, search for oxidation on methionine, things like that. And from that, these kind of orange spectra here will produce your identified peptides and your identified proteins that you know are in your database. You then create the subset database, which is much smaller, making your computation much quicker. You can then repeat the pass on the analysis on all of these unidentified spectra represented in blue that probably come from these same proteins, but perhaps weren't fully tryptic due to poor enzyme activity. Or maybe they had a PTM that you had not considered in your initial search. You can now search for them now. And this is a way of actually getting through very complex searches with lots of parameters in a very quick fashion using this multi-stage approach. And so XTandem does it really well, and you might see it in other algorithms. The other thing that makes XTandem somewhat unique is it has different scoring functions that you can plug in and out to make it look like another algorithm, such as this k-score, which makes it look like sequest. But also there are other scoring functions that are provided in the package. But each of these can actually change the function and actually create almost like if you were to repeat your searches using each of these different scoring functions here. It's actually like using 3 different scoring algorithms, 3 different search algorithms to search your data, which when we learn about the profits, and in particular iProfit, this is a great advantage for you mathematically in identifying more spectra, more peptides from your spectra. And this just goes into the k-score plugin, which replicates to some extent the sequest scoring algorithm, which I had already discussed. One thing that's important to note within the TPP, this is an actual image of the peptide PepXML viewer, the peptide spectrum matches inside the TPP, is that when you use these alternative algorithms with tandem, these alternative scoring algorithms, that it actually replaces the tandem's original score with whatever algorithm you use. So it's important to know that the scores are modified in your results. Comparing the best score in a spectrum to all the other scores that are possible for all the other peptides that were searched against that spectrum. And so if you have, for example, our 1000 Dalton peptide that had over 1,000 different possible peptide matches to it, and you were to plot a distribution of all of those scores, it would look something like this, where there would be a lot of peptides would have a very poor score, but only a few would have good scores, and perhaps only one would have a really good score. So if we go back to this slide that shows the best score for a given peptide spectrum match and the next best here, they're represented as the best score down here and the next best over here. What you would want for a good score metric is one that separates this score here at the upper end of the distribution as far away from as possible from all the other peptide scores. And so this is what we have, our algorithms that the TPP uses that will help determine what the best relationship is and how to obtain the best score and what the cutoffs are from the rest of the distribution in order to accept a result. Regarding other search engines, we have some additional ones listed here. Of course, Comet, my favorite. There's OMSA, Inspect, Mirror Match, Phoenix, Spectrumill, Protein Pilot, etc. These are all search engines that you might see. Each has its own unique features, advantages, and disadvantages. Some might be better with particular PTMs. Some might be faster than others. Some might have more comprehensive scoring algorithms, and so will consistently produce more PSMs than another algorithm. But those PSMs might be different than another algorithm, and thus would facilitate the need to use multiple algorithms to find the maximum set of peptides you can identify. The big question a lot of people have is, what is the right tool for you? And this is a very good question to ask yourself. There's some other questions that can help you narrow down which is the right tool for you. And the first being is, which can you afford? If you have a $0 budget for doing your computational analysis, Comet is your algorithm right there. It's free, so use it. Tandem's another good one. I think Mirror Match is also free, and OMSA are free. They're also, so a lot of people might be limited by their costs. Can you easily get to the results you're looking for? And that's important. With different algorithms, if they output the results in formats that are incomprehensible to use without some third-party tool to interpret them, then they're probably not right for you. Everything that we have in the TPP boils down to standard data formats, and so they're actually interchangeable between the components of the TPP and other algorithms that support those same data formats. And so it's important to choose tools that provide results in with good data integration. Actually, that's actually another point down here. And then, is it compatible with other tools is very important as well. Mascot is kind of a good example as well. If you have Mascot, and it's great for identifying peptides, but if you also want to quantify those peptides and quantify your proteins, Mascot has a tool for that in its pipeline, and so it's good to do that. It would be a good tool for you if that was your interest. The TPP as well has several quantification algorithms that might be right for you, and so consider these additional tools that are required for your analysis. And then the last part of this tutorial here, in the last 5 or 10 minutes, I'm going to talk about database search validation. And the way that we primarily do this is what's known as decoy databases. And so what are these decoy databases? Well, inside an actual protein database that you have are all the possible proteins that you expect to find in your sample. You expect to see all of those. You would hope to see all of those proteins. What you don't know is whenever you identify a peptide from that database is whether or not it's correct. If you provide in that database an equal number of protein sequences that are all known to be false, then you know identifying any of those peptides produces a false identification. That false identification is enormously helpful in estimating what your false discovery rate is on your entire sample. So there's what is usually done with the use of decoy databases is you search simultaneously a set of target and decoy protein sequences. You can possibly search these sequences independently, but then you have to combine the analysis downstream at a later time. So usually we analyze everything together. It's much easier that way. Decoys can be generated in multiple ways. One of the simplest ways of doing it is taking all of your protein sequences and just reversing the amino acids. So they all start with methionine and then maybe alanine. Well, you reverse all the amino acids to the opposite, and then all of your proteins will end in methionine. That reverse sequence usually produces a unique set of peptides when using a trypsin constraint that would not be seen on a forward read of your database. The other way to do it is, that's commonly used, is to shuffle the database or just kind of randomly reorganize all of the amino acids in a protein sequence. And it's very unlikely mathematically to randomize them into peptides that you would see in the unrandomized sequence. These decoy matches are used to estimate false discovery rate. Any match to your target database, you don't know if it's real or false, but every match to your decoy database you know is false. And so you can estimate you can estimate your false discovery rate by counting the number of decoy matches in your results and dividing by the number of target matches in your results. And so a lot of people ask, well, what score cutoff should you apply? Well, what you can do is rank all of your peptide spectrum matches in order of most probable to least probable. And then your decoys will be interspersed among there so that as you step along and count the number of target hits you have, you can produce, in this case, a Q value, or an estimate of the false discovery rate along here on the x-axis by saying that over 4,000 target hits occurred before that first decoy was found, and that eventually you'll have enough decoys to come to a 1% false discovery rate, at which point you've identified maybe 5,000 or so target peptides. And so you can continue monitoring this, and if you increase your false discovery rate, you do get additional target hits, but there's a consequence to it. It's, first of all, diminishing returns. You're not getting many more target hits the higher you go in your false discovery rate, because eventually you just start incorporating so many of these decoys into your analysis that you're not actually able to pull in many new triptic or target peptides. In addition, this FDR value is across all of your PSMs. So if you have 4,000 target hits before your decoy, you are fairly confident that all, or maybe almost all, remember decoys are just an estimate, are real hits. But if you come out here and you accept 6,000 target hits, but at a 5% false discovery rate, then 5% of that entire 6,000 is false, but you don't know which ones are false. And so in this case, it might be better to only accept the 4,000 or so hits that you know are highly confident, rather than to reduce your confidence in those 4,000 hits just to obtain a few more. So some additional validations that can be performed is accurate precursor mass measurement. This is important, especially if you use instruments such as Fourier transform or time of flight instruments that can measure very accurately the precursor mass. That by specifying a very narrow peptide or precursor mass, you can eliminate false positives if they're just outside the mass tolerance of the instrument. There is a small caveat to this, and that is we usually trust the instrument to identify the precursor mass accurately, but occasionally it identifies one of the isotope peaks as the precursor mass in that peptide. And when performing the search, you don't actually get the right sequence until you tell the search algorithm to adjust for this precursor mass by adding a Dalton or 2 Daltons to the presumed mass, or actually it's usually subtracting a Dalton or 2 Daltons from the presumed mass. And so there are parameters that can handle that as well, and that you will learn about in particular with, say, the Comet search engine. There's also some general rules to consider when interpreting the results. And the first is if you actually performed a triptic digest of your sample, but then did an unconstrained search, you would expect that the majority of the correct answers, the correct peptide spectrum matches here, would actually be to triptic peptides. So peptides that are preceded by a lysine or arginine and end in a lysine or arginine. You might expect some of these semi-triptic peptides to occur, such as this one here, where on the N-terminus, this is triptic specificity, but perhaps due to the length of this peptide, there was a breakpoint here that occurred, creating a non-triptic C-terminus. You might expect to see these, but if you look at your results and the bulk of your results come back as being non-triptic, then I would be very skeptical of those results because you expected triptic specificity given your experimental design. So consider your experimental design and your enzyme as well, and this is a way that you can very quickly evaluate your results. Another thing you can do, and the TPP comes with a viewer known as the lorikeet viewer. You can visualize each of your spectra and determine how correct those PSMs are just based on the number of fragment ions that match to the major peaks in the spectrum and the percent of the peptide represented by peaks in the spectrum. And so correct matches will actually contain these very nice B ion series and Y ion series, very robustly represented, and all of the tall peaks are generally identified in the results if you were to visualize them. Even in spectra less dense than the one previously shown here, there are certain indicators that might be important. Like typically, Y-ion from an N-terminal cleavage of a proline produces a strong peak. And so you expect to see this enormous Y-ion peak here that happens to represent a proline over here at the Y7 ion. On the other side of it, the B ion tends to be missing in these cases. and indeed there is a gap with the BI in there. So you can use simple interpretation rules such as this proline rule to evaluate your spectrum. And then this actually wraps up the lecture for this part here. All right. Well, thank you.
So the one thing that we try to make sure that within the TPP is that we have a capability of converting instrument raw files into a standardized format, and then you have this capability of then using those throughout the TPP, and the results from each of the tools are also converted into a standardized format, so they can talk to each other, and it's easy to then to plug in different tools and do that. So this is where we are at the moment. We're going to talk about the data formats and also the PEP3D at this time as well, in which you can start to visualize the data once you've done your initial search and actually have a look at what all the spectrum looks like. So Luis Mendoza will be the one that's going to take you through this. Now, Luis is your primary software developer for the interface, things Petunia that you'll get to start to use. He's very involved in making sure all these tools interoperate with each other. And so he's the guy that you go to when something breaks in the middle and says it doesn't work, and he usually says, well, you've done something wrong. But apart from that, he'll take you through that. So where is that component? And so we know that we have this trans-proteomic pipeline, and really we have a couple of areas where these data formats come into it. The first one is a data format here, which is what we started off doing is MZXML, is now MZML. These are all different formats as they've been produced. At the start of the TPP, we were producing our own tools for each separate vendor. This has now been taken over by a consortia which has got together called ProteoWizard and use MS Convert, and that brings that into here. But the other formats that Louise will talk about is how once you go from the raw data through the database searching that Mike just told you about into the results section, that all of these tools here can then have a common format at the back end so that these tools can then operate with each other, and then the output of those can then get moved along that pipeline. So you have this consistent capability without loss of any data. And so this is just a brief list of the formats. The most current one is the MZML, but I'm not too sure if you're going to talk about even further ones, MZ5 or anything like that. No, at this point. But we also there's a number of different tools that are being produced for very large raw files that are coming from the data independent analysis that also need to be considered too. And so this is what we use primarily at the moment. We use the MS Convert for those. And then the PEP XMLs is what is used in between the database searching and where all the peptide matches are stored. And then you have the Prot XML, which Louise will go through, that is then the output of the protein profit. And so why do we need these formats? And so you can see different instruments, they look different, they operate different, and of course the software has to be different. And that's one of the biggest problems in the industry, is that no one can interoperate with each other. They all like to do their own thing. They say that the data format has to be different because that's the way that the ions are stored as part of the detection system. But there is those capabilities. It's been proven that you don't need to have all of that information as a proprietary component, but yet we still persist with this difference between all the different vendors. And then when you come back at looking at the software, it can get quite expensive if you're buying software from one vendor, and then you have to buy another piece of software from another vendor, and then a third vendor, depending on the instruments you get. Or you can buy an outside third-party software tool, which can be also expensive. And you'll find that a lot of those third-party vendor tools are actually based on the TPP anyway. And so if they're doing that, then if you see things like peptide profit in there, you know that the TPP is the background of that, that there are also versions behind of what we have as the current version. So even though they might have a slick interface, you know that the tools are slightly outdated. And then these open formats enable you to do this sharing between the instruments, between the labs, normalize scores across data sets and apply this common processing pipeline. So that's really the basis of where things are. And so Luis is going to take you through all of those now, and then we'll continue on from there after lunch. How many don't have a stick at the moment? Count them and make sure we have them tomorrow.
Here I'm just highlighting the place where the data formats live. So the TPP Trans-proteomic pipeline is, I guess we haven't mentioned this explicitly, but it's something called open source and free collection of tools, but also data formats. So free means exactly the word free. You didn't pay a dime, I guess, to get TPP, and that's great. I mean, we all like free. But this term open source, it means that in the context of the software itself, all of the source code, how everything is implemented, it's an open book. You can, if you know how to read computer code, you can actually read all of how the TPP is put together. It gives you access to question and evaluate the all the assumptions and all the statistical models within it. But also, if you have the expertise, you can change it to your own liking and have your own custom version of the one component TPP. So that's actually great. Other software out there, obviously, you have to pay for, and it's black box. So TPP is great, we think, because it allows you to do both of those things. But it's not just the tools, it's the data formats. So we're going to talk about those, and this is just an overview, and Rob showed this as well. So we have the tools here on the left side, some of the tools we have in TPP, but also these data formats. All of these formats, at least in the computer speak, are based on this meta format called XML. And XML is just something that stands for Extensible Markup Language, and allows you to create documents, computer documents that have this self-describing format. And XML consists, any type of XML consists of text, or so just a text format. It's not a binary format, organized within tags. It's supposed to be, as we call it, human readable. You can actually look at some XML code and sort of figure out what it's trying to represent. It also makes it very easily machine readable. So you can write fairly quickly software that can read that format and process it and extract information that it needs. So this is just a very simple example. You don't need to be a computer scientist to sort of figure out that this snippet of code perhaps represents something like a library entry, right? So there's a book with a certain type, science fiction, sci-fi, location, it's perhaps where it's, what part of the library this is located, and there's some author and title. You could replace these tags with, scan number equals something, and type equals MS or MS2 or something like that. Collision energy equals some amount, and fragmentation type CID, and then peaks, M over Z, and so on. So you could see how you could represent those types of data in this format. So we have right now in TPP 3 formats, and the first one is this MCML format, and these other two I'll talk about in a second. So MCML actually started being, we've changed the name a couple times for various reasons. I'll maybe just gloss over it here a little bit. It used to be called MCXML. And these MCML and MCXML are formats that decouple your spectral library, your spectral data from relying on your vendor format. There are multiple of these. Older incarnations, there was something called MCData a long time ago, and MCXML. The current sort of standard format, Rob mentioned the PSI, the Proteome Standards Initiative of HUPO, has come up with this MCML format at version 1.1. That's the latest format. And this actually took years of integration between the HUPO PSI initiative, those of us at ISB who have developed MCXML originally, actual software, machine vendors, and some other people. And the funny thing is, it actually took maybe less than a year to come up with the actual format, and then 3 or 4 years for the actual lawyers of this, the legal departments to agree to actually do this. So, it's they probably even made money doing this. We didn't make any money doing this. So specifically MCML then. So that's just a little bit, just a one slide of history. But MCML is the standard data format for representing mass spec-based proteomics. Also, it's been extended to represent metabolomics, so anything having to do with some mass spectrometry data, at least in our fields here. As I say, it was a joint effort between the HUPO, Proteomics Standards Initiative, us, and some software houses and vendors. The design of MZML is this XML that I just showed you a little while ago, but we also incorporate something called this controlled vocabulary. Controlled vocabulary is basically a dictionary of words of words in an ontology that very thoroughly define the terms that you're going to use. Perhaps some of you have used some online, querying tools. You're looking for a very specific protein, and you can see it written down in many different ways. For example, trypsin with just lowercase, or trypsin with a capital T, or trypsin and then there's a dash or something. And so, if you're trying to search for this, it's kind of hard to find the exact right, correct thing. In gene ontology, this gets very crazy. And so by having a dictionary, a reference of words that you're supposed to use, you are able to reference the exact instrument type, the exact fragmentation type, the exact, enzyme that you're using with a central, via a central reference. So this is sort of the big thing that MCML incorporated. But it's flexible enough that it allows inclusion of new knowledge without having to change the format. So when, SciEx releases a new instrument, we just have to add, we don't have to change the format, we just have to add to the ontology, to the central, dictionary, a new entry for that new, mass analyzer, and it will just be part of the reference. So it has survived quite well. I guess this format has been around for about 7 years now, and it's we haven't had a need to change it too much. What this does create, unfortunately, is an extra step in the analysis of having to take your original data file, and in whatever format it is, and converting it to this MCML. So we do have now this new platform that Rob mentioned. It's called MS Convert. I'll show you this in a second. But we basically take data from any number of machine vendors and run this one program that converts it to an MCML file. And that still tends to be, unfortunately, the barrier of entry into this whole open source. You require certain software to be installed in your machine, and sometimes this just doesn't go extremely smoothly, but we're trying to get to it. So for this first step, the available converters, the only one we use nowadays is this thing called MS Convert. MS Convert comes from a third party, and so again, this is the beauty of open source and having these open source licenses. We didn't write this MS Convert software. This our colleagues at ProteoWizard project did it, but we're allowed to include in TPP and redistribute it. Just like TPP, you can incorporate into your own software and redistribute it as well. And so, so this handles conversion from several software, instrument vendors. I have a list on the next slide. You can also, to make, one of the unfortunate side effects of having this XML type format is that the original data becomes larger when you do XML. You're adding many words to it, and it's text-based. But you're able, if you want to, if disk space is an issue, you can actually compress your peak list or even gzip the entire file. And as far as TPP goes, it doesn't even care that it's gzip. You can just, all the tools will run against it. It's just slower because you have to decompress that every time you access it. But otherwise, the tools don't care that it's compressed. But you'll just suffer, in performance-wise. I just include this in this grayed out area. Some of the old software that we used to include in TPP to convert very specific instrument vendors to MCXML, but we don't use this anymore. We don't support them. But you might find, if you do a web search, you might find some of these names out there. And they sort of still work with older data, but they will not work with newer data just because they weren't coded to do that. we only use MS Convert nowadays. So this is MS Convert, again from ProWizard. These are the vendor formats they support as of, I don't know, a couple of weeks ago when I just went to their webpage, this webpage, and this is the verbatim from their page. So they support Agilent, Bruker, Thermo, Waters, and I guess Applied Biosystems, that's called what? SciEx, right? They haven't changed the name on this. SciEx instruments. And all the softwares and the licenses are distributed with the with MS Convert, and it's including TPP. That being said, it's hard to control. By the way, this only runs on Windows systems. So the deal that came out of talking to all these different people and their legal teams and their intellectual property was that they could only do it with Windows, and they are the ones that provide something called a DLL, basically a library, a dynamic link library within Windows that allows MS Convert to access those raw files that they produce. And that's only in Windows. So even though TPP is actually developed in Linux, and we also distribute a Windows version, in order to do the conversion, you have to use a Windows machine. Not only that, some Windows versions, depending on which specific Windows version you have, and there are many, may not have the exact libraries, like .NET. This is one of those Windows things. Very specific .NET libraries are required for these different vendors to run. So it may be that your computer, even with the software we gave you, may not perfectly run. You have to go to Microsoft, and they have a page where they tell you what you need to install. It's all free, but you may need to jump that extra hoop to actually get this to run. That's just unfortunate. They could have done it a different way, but this is the way they chose to give us access to their data. So, okay. Any questions so far with MCML? So that's the converted part, visualizing MCML data, and you'll see this in a later lecture. We have this software called Pep3D, and I'll talk about it in the, I think, in the next lecture. Now, so you got from having some raw data now to a common data format. Now you run a search engine, Comet or Sequest or Mascot. Well, unfortunately, they produce their own format, output format. So we came up with another format to specify peptide data, and it's called PepXML. So PepXML is the standard format for representing searched data, so search results, peptide, PSM results. It stores the format itself. It's also an XML type. So instead of having scan number or something, you probably have, peptide match, X and score, X score equals something, so the search engine output. You can store that information about these peptide assignments to the spectra from a number of search engines. We support about a dozen nowadays. I actually can't keep track because we just really use 3 or 4 at ISB. But we have about a dozen search engines that we support in TPP. You're able to combine multiple MS runs. So if you did, say, like a fractionation or just a repeated experiment, and you want to analyze it in one go, you can actually just combine those 2 or 3 or 4, 10 run, or any number of runs into just one analysis. So you're able to combine, to store several search results into, from multiple MS runs into one. It also goes beyond that. So this format also allows you, at the peptide level, to store validation results. So not just the search engine result, but what we're going to learn about later in today, the peptide profit or any other sort of peptide validation step. It can also, this format stores that as well as peptide level quantitation. And we'll talk about quantitation tomorrow. And again, because you can store multiple MS run results, you can also, then this can reference one or more MCML files, because one MCML file is one run. This does create a need to have converters for certain search engines that do not just produce PepXML. So for example, we have this software called Mascot to XML, which will take a .dat, I think this is called an intermediate file in Mascot. I don't know, I don't really use Mascot too much. But you can take a DAT file and convert this to PepXML. For Sequest, we have another conversion. For Tandem, we have another converter. Fortunately for us now, more modern search engines such as Comet can just write PepXML directly. So we don't need to do anything to their output. We just use that as an input to our other tools. So PepXML is just not a format. We have ways to convert into that format as well. So these slides, we have them so that you have them as a reference later on. So we have a question about PepXML, you can actually look at this slide and say, oh, I wonder which kinds of tools write PepXML. So these are some of the tools that produce PepXML, so the converters. Some of the search tools like Comet and Spectrast that you'll learn about actually this afternoon. And also the validation tools like Peptide Profit, iProfit, PTM Profit. I should add that there. And quantitation tools at the peptide level. And there's things that read PepXML, such as the visualization tools, some of the validation tools, and other tools that we have. To visualize PepXML data, we have a PepXML viewer that we'll be actually using quite a bit in the tutorials later in the week. And finally, ProtXML. So you can tell by the name, it gives it away. So this is a format for representing identified proteins, so not just peptides. So ProtXML was developed as a way to store protein profit validation results. So at this point, you don't have search engine scores. We have, as you'll see when we talk about the peptide validation, at some point you have validated the peptide, and now you just want to store not every single spectrum, not every single search score, but just what is the evidence for at the protein level, which peptides were part of that and what scores they had, like probabilities from the validation scores, but not the search engine. So this creates a much smaller file that you can share and analyze and so on. Otherwise, the PepXML files get very big, especially nowadays with modern instruments. So ProtXML stores protein profit validation, but also quantitation at the protein level. It can reference one or multiple PepXML files, and once you have all that information there, you can import this into some other maybe archiving systems where you want to store your identified protein data, or some other tools out on the web that take this as an input. One thing to note is that as much as TPP is used and actually has a widespread adoption around the world, really, in our community, a lot of people actually use just our formats because they want to, someone maybe writing a tool for their, PhD thesis or maybe a postdoc project. And if you write a tool that can read and write either PepXML or ProtXML, depending on what the tool you're using, you can immediately just plug it into TPP and it'll just work with all these search engines and all these other tools downstream. It's the quickest way you can get your software to actually work with a bunch of other software. If you just, most people just write software that will produce some tab delimited, output. And then what do you do with that? You have to know which column you want to incorporate. So it's actually funny to see when I go to conferences, see how many people use PepXML as their base, because they're immediately connected to a dozen search engines and a bunch of other validation tools. So it's kind of nice to also have that, I guess. So there's readers and writers of ProXML. Here's a list of some of these that we have in TPP. We have a visualizer for ProXML, and actually we are now mentioned MCML. And so how do you view an MCML file? Well, I guess you could open a text editor because it is a text file, but most people will not want to do that. They want to actually view it. And so we have this software called Pep3D. I guess I should turn this on. Pep3D. And so it sort of sits in this pipeline here, so it sits here between the raw data and also the validated results. You can overlay information from that as well. Why visualize LCMS data? Well, a single spectrum, if you have a fragmentation spectrum, you could see, well, what happened there, at that one specific instance, at that snapshot in time. And those spectra, tandem spectra, lead to the identifications, but typically only the most abundant signals are the ones that are examined and analyzed. But what else is occurring in the whole run, right? So if you want a more holistic view, you want to look at the entire LC-MS space. And so, visualizing this is sort of the most simple way of doing it. You can also just do statistics and have, some other quality measures for a run. But sometimes looking at it is just nice as well. Does Mike show this slide? And see, this is just when you take slices over time or scan number of the different MS1 spectra, and over time, and you can see, how the intensity changes over time. This chromatogram here, but you can then just put this in. These are 3 dimensions, right? Time, m/z, and then the intensity at any given spot. So you can get something here that looks maybe something like a gel. It's not really a gel, but you see the chromatographic retention time here on the x-axis, mass to charge ratio here on the y-axis, m/z, and then the darkness tells you just how much signal was measured at at any given point. And why is this useful? Well, you can see, and then for this software, you can overlay not just the raw data, but you can overlay CID. So there's within the software, you'll see there's a little checkbox or a little drop box where you can select CIDs, and it will color all the events that led to a chart, to a CID event, to an MS-MS spectrum being produced. Turns out all of these things, if you click on any of these things, you'll get a spectrum. I'll show in the next slide. Also, you can overlay, if you did already a search or even a peptide profit run, you can overlay the probability on that. And so you can see where the good hits came from and maybe the bad hits, and gives you a count also as well. And you can do a cutoff of what probability cutoff you want to display. Or all of them. You can display all of them so you can see what ratio of well-identified peptides over all the CIDs you got on this one run. And also, each of those things is a link that takes you to just the MS-MS spectrum, so you can just view without this won't be interpreted. It's just the raw spectrum, so you can just see if it's a high quality or noisy or it has a lot of peaks or not too many peaks. A little bit hard to see on this display, but probably if you're following along on your laptops, you can see this better. You can see a lot of peaks there. And you can play with this spectrum viewer, and we'll be using that for the tutorials as well. And so how can you use this software? Well, you typically, especially for those of you who are experts already at mass spectrometry, you probably know this more than I do. But you typically want to see this, plenty of well-localized spots without any large-scale pattern, so in the solution, and gives you good separation and good opportunity then for the mass spec to sort of isolate and get these things sequenced. Unfortunately, sometimes you maybe get something like this where you don't really see a sample. You have very few spots in your entire run. You mostly get background noise, and you can't even distinguish this from a nose spray. It will be very difficult to expect good results when your data are this bad. And sure enough, when you do the analysis, you see that you actually, out of the 300 CID events, none of them really produced a good peptide, at least in this one specific example. Sometimes you can tell perhaps if your separation was not optimal. In this very specific case here, you can see that the entire sample here is in a very narrow zone of the elution gradient, only between maybe 60 and 120 minutes, but you acquired for over 3 hours. So you're just wasting instrument time. Many intense ions are not fragmented. You're just not giving enough opportunity for the mass spec to go and get all these things. And so you may want to then, upon looking at this, maybe change the way you're fractionating or putting your buffer into this. Pep3D does not give you this red line. This is just for comparison. This is just to show you how this gradient was applied. And again, you see a very empty space here for the first hour and a half. You're just running your machine, but there's nothing being produced for over an hour. Why would you want to do that, right? And you see that at the end, you kept trying to acquire things, but there was really no data. All these horizontal streaks, they're really not peptides, and you can tell that they didn't score very highly. This, again, when they did this many years ago, they actually ran the exact same sample on 2 different columns. One was, I guess, an older, just dirty, overused column, but otherwise the exact same sample went in, and you can tell the difference just by, just looking at it. This is very noisy, has all these streaks everywhere, and you can see that not just the CID, you got, almost 40% fewer CID, or 37% less IDs, but also if you try to do quantitation, which we'll learn about tomorrow, but it will be much noisier and you'll suffer from the trying to do quantitation or something like that. So again, visualizing it helps you see what the quality of your data is. You can also look for things like chemical contamination. If you're trying to acquire, if the instrument is trying to fragment things that are very low M over C, especially like if singly charged ions, of course they will not really lead to very good IDs from your database, or you're just wasting instrument time. You're wasting CID attempts on something that really, maybe you should go back and look at your entire system and clean this out before you maybe do more experiments with this setup. Again, other contaminations